fungi and the matrix in animals. So we saw that there are sort of two kinds of bacterial cells, the gram-positive type. These would look purple under a microscope after the staining technique, have a thick peptidic glycan layer. And we saw that that is composed of polymers of two alternating sugars, N-acetyluronic acid, NAM, and N-acetylglucosamine, NAG. And then in order to cross-link these polymers, that's where the protein part comes in. Attached to the NAM molecules are 4 amino acids. That's the 4 blue circles. And the purple cross-link is usually 5 glycines, but it can also represent a direct simple covalent bond between them depending on the exact cell type. So it's just layer upon layer of this kind of situation to create this thick peptidic glycan layer. And then we saw the teichoic acid. These vertical kind of elements here are sort of reinforcing type of elements. And then the version with the lipid attached seems to insert itself into the cell membrane and basically seems to anchor or connect the cell wall to the cell membrane across that little periplasmic space. And then we saw the gram-negative situation. These would be pink at the end of the staining process. Have a peptidic glycan layer, but it's much thinner, and probably because it's thinner, it doesn't have the teichoic acid reinforcing rods, but it does have an extra membrane called the outer membrane. And we saw in the outer leaflet of the outer membrane this sort of weird glycolipid called LPS, lipopolysaccharide, that we saw was is an endotoxin. So as you kill off these bacteria by taking, say, an antibiotic or something like that, as they disintegrate, as they break apart, they release this, and that causes low-grade fever and some other aches and pains kind of idea. And I think now that I look at this, I think I forgot to mention last time, this last protein here, this bronze or Brown's lipoprotein, I don't think I mentioned that last time. As you can see, sort of links the peptidic glycan layer to the outer membrane. So that's the linkage here. Okay, then we looked at the fungal version, which is very, very similar in sort of overall structure to the gram-positive bacterial cell membrane, except instead of having polymers of 2 sugars together, we have 2 sort of different polymers here of 2 different sugars, glucan and chitin are the 2 polysaccharide components, and the proteins that are going to cross-link the carbohydrate polymers is this nanoprotein here. So again, very much like the peptidic glycan layer of the gram-positive cell, except we swap out different sugars and different proteins. That's going to be kind of the theme. And then in the plants, we have, of course, cellulose as the major polysaccharide, along with glycan, sort of related to glucan in fungi, and then the major protein is this pectin. Again, it's always sort of just a combination of sugars and proteins, so you just need to keep straight which sugars and which proteins go with what organism's cell wall. And then we looked at the extracellular matrix of animals. Animals, of course, don't have a cell wall, but we do have a layer outside our cells called the extracellular matrix. So we're going to focus on sort of this type in connective tissue. And we saw a number of different protein elements, some of which have sugars attached. So it's the same sort of overall theme, but the big difference, of course, is while the cell wall protects from osmotic lysis, the extracellular matrix doesn't do that. It's sort of just a glue to keep cells together in tissues. So we saw collagen as a major component. We saw elastin, proteoglycans, which we had studied back in probably September, fibronectin, the basal lamina specifically made up in large part of laminins. Again, be careful with that name. And integrins, we'll take a closer look at these proteins today. In these hemidesmosomes and focal adhesions. And then here's sort of our put it all together picture. So we have collagen fibers and proteoglycans and fibronectin, and they're not showing necessarily everything in here, but elastin would be in here, and all the other proteins that we saw, including these integrins. Again, more on that today. Linking sort of this is sort of the transition between the extracellular matrix across the integrin, that's the integral membrane protein, to the cell cytoskeleton on the inside that we looked at earlier in the week. Okay, and that's where we got to, I think, last time. How are we feeling about cell walls and the extracellular matrix? Nothing to explain, just one of the proteins of the extracellular matrix. Okay, final part of this topic that will again connect back to parts of the cytoskeleton and those integrin proteins that we were just looking at are junctional complexes. So specific spots along the membrane, and we're going to focus on epithelial cells that keep cells attached to each other in an epithelial sheath. So think your skin, the mucous membranes, those kinds of ideas. So we're going to see that there are a number of them. We'll kind of work our way from kind of top to bottom here. So this one on the left is the figure from your book, which kind of, again, is a nice little summary table. So we'll be looking at things called tight junctions and adherens junctions and desmosomes and hemidesmosomes and gap junctions as we go through today. Okay, so you can see that they're sort of grouped in different locations along the membrane, on the sides and the bottom of the membrane. And then this is sort of the same idea, just another figure from a different book showing the same types of junctional complexes that we'll be looking at. Another sort of summary table, if you want to take a look at this towards the end as a sort of a quick review. Okay, I do like to show at least some evidence for all of this. So again, we have lots of good microscopic evidence for all of this. We're not just making this up. So we have all types of junctional complexes that we can see under the electron microscope. So tight junctions, adherens junctions, desmosomes, gap junctions. So there is very, very good microscopic evidence for everything we're going to talk about today. Okay, we kind of start sort of towards the top of the cell, or at least the top on the side. We have tight junctions. And as their name sort of suggests, these are going to very, very tightly connect to cells in an epithelial sheath. So you typically find them kind of towards the top here. So these would be the tight junctions. The major protein that makes these up is called a occludin. Protein, and as you can kind of get the feel for here, it's a large number of these proteins, so it's an integral membrane protein, right, that comes sort of sticking out of the cell membrane on both sides, on the two adjacent cells, and basically the best analogy I can give you is they sort of act like Velcro. So they sort of hook on to each other, and just like Velcro, the more Velcro you have connected to each other, the stronger the connection is. These work sort of the same way. So the more occludin you have in the membrane that are sort of hooked up with each other, the tighter and the stronger that connection is going to be between the two adjacent cells. And this top figure is pretty good as well because it kind of gives a quasi three-dimensional idea here. So you need to picture this as a ring around the entire cell, right? Because if we, I'm not even going to attempt to try and draw that three-dimensionally, but you need to picture these cells. If we kind of looked at the sheet from the top, here's our central cell. Here are cells on all sides, right? That we would need to connect. So this this occludent protein does go all the way around, so that the cells are connected on, sort of all sides. Kind of picture what I'm talking about in my terrible drawings. And it does go all the way around, so it is connected very, very tightly, as the name suggests, to the cells on, all sides of it. And tight junctions are incredibly important in epithelial sheets. The really, really good example will be your intestinal epithelial sheets. So think about your intestinal cells, right? So here's your intestinal epithelial cells. Here's your intestinal tract where all your food is, right? When you just consumed your bagel for breakfast or whatever. And here's the barrier basically between the outside, in a sense, your food and stuff. And so down here's your circulatory system and things like that. You really don't want all the stuff that's in your intestinal tract to just have complete access to the inside of your body. Right? You don't want everything here necessarily jumping right into your circulatory system. You want to control that. Right? You want to control what you absorb out of the food and what you don't. And in large part, that's what these tight junctions enable your cells to kind of set up. They're a barrier. They're a permeability barrier. So this is a good example here. In this experiment, they've flooded one side with what they're calling your tracer molecules, some kind of, say, fluorescent molecule or something that you can see. And in this epithelial sheet, where each cell is sort of connected to the next by these tight junctions, those tracer molecules can't get across those tight junctions. So the molecules can't sort of leak in between adjacent epithelial cells. So if that molecule needs to get from here into your circulatory system, it's going to have to be absorbed by that cell and then sort of re-secreted or released on the other side to release it into your bloodstream. So that means your cells, these epithelial cells, have control over what's going to be absorbed and what isn't from the bagel or the donut or the steak or whatever you had for breakfast or dinner last night. So it's a really important idea in terms of presenting a barrier. Same thing with your skin. You don't want everything from the outside environment just leaking in between your skin cells, right? That'd be really, really bad. So these tight junctions don't allow that to happen. They're a permeability barrier. And you can see it works both ways. So if you put the dye up here, it doesn't get across into the space below the tight junctions. If you put the dye on the other side, it doesn't get across the tight junctions out into your intestinal tract. So you don't want things leaking the other way either. Okay, so it's a really nice barrier to keep things, sort of on their own side kind of idea. Think about it as the fence between neighboring yards, right? You don't want your neighbor's stuff just willy-nilly coming into your yard, and your neighbor doesn't want you coming willy-nilly into his yard, so this is kind of that kind of barrier between them. So these occludent proteins then bring the two cell membranes really, 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 really close together. And probably a few more reallys. Right? However, it's now been clearly shown that the two membranes are not fused together. When I first learned about tight junctions back in graduate school 20 years ago, there was still the idea, in fact that was the idea, that the two membranes had actually been fused together by whatever the protein was. They didn't even know what the protein was then. But it's been clearly shown now that the two cell membranes are not fused together. That they're basically as close together as they possibly can be without being actually fused together. There's a lot of really good experimental evidence to show that they're not actually connected together. But they are very, 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 very close together.
They're literally just they're not touching at all?
Oh no, they probably are touching, but they're not fused together. Meaning they're still separate membranes. It's not like that, where the two membranes have been fused together. So that's not true.
Does that pose any stress on them though? Any kind of stress?
Stress. Define stress?
As in, well, like the pressure of just the two things going against each other, does it make them, I don't know, do they react from that, from just being pushed together like that in any kind of way? Or being stuck together like that for a long period of time?
I'm going to say no, since I've never had that question before, so I've never seen anything that would suggest anything like that. So I'm going to say no. Or at least not to my knowledge. That'll be the safer answer. Okay, so that's a little bit about tight junctions. So they're basically, like I said, a permeability barrier. So nothing gets across this way, nothing leaks out the other way across a tight junction. So just another version of that kind of experiment. So again, you put the dye on this side, they can reach up into there, but can't cross that barrier of the tight junction. So the tight junction must be right there. Okay. Usually a little bit further down, sort of on the side. Actually, before we leave tight junctions, one other thing. Not only can things not leak in between, if you have a membrane protein, say up there, blue one, versus, say, the green one down here. When we talked about membranes, we said most of the proteins can, sort of just float around and move around inside the membrane, and that's true, except at these tight junctions. So that blue protein could diffuse down, but can't get past the tight junctions either. That green one could go up to here, but not get past that tight junction either. So tight junctions also actually define, they're usually referred to as domains of the cell. So the part above the tight junction is usually called the apical domain, and the part below the tight junction is called the basolateral domain. And they are separated by those tight junctions as well. So that's another thing sort of the cell needs to keep aware of, in that if that blue protein needs to be over here, then it's got to send that vesicle to the apical domain and not the basolateral domain, because if it sticks it down here, it can't diffuse upwards. Same thing with the green one. If that needs to be down in the basolateral, it's got to go there first and not be directed to the apical layer or apical domain. And so it's another sort of little, not twist, but another little detail about making sure those vesicles are being directed to the right location. So presumably there are going to be snares specifically for the apical versus the basolateral domain to get those vesicles with the right protein to the right place. So it's quite the formidable barrier. Less so, or less of a barrier like that, are things called desmosomes. So these are usually a little bit further down, as you were seeing in those earlier pictures, and these are not nearly as tight as the tight junction, so things can cross that space, so they're not doing the same thing. But these just more kind of in general link adjacent cells together in a sheet, in an epithelial sheet especially. And desmosomes are going to include intermediate filaments, so these are the sort of junctional complexes that allow that stress. When we were talking about intermediate filaments either earlier this week or late last week, when we press on your skin, it doesn't fly apart because of intermediate filaments. So that pressure would come along these intermediate filaments, cross this desmosome, and sort of be distributed across the next set of intermediate filaments in the next cell. So these intermediate filaments sort of work with these things called desmosomes, which are composed of these two pieces. There are a bunch of proteins that sort of collect here that are referred to simply as a plaque. But this is a collection of proteins. This is not just one big thing. It's an aggregation of smaller proteins, but people still refer to it simply as a plaque. And then sort of the hooks in between are proteins called cadherins. So these little hooks in between are called cadherins. So it's sort of a three-piece complex here for a desmosome. So intermediate filaments, plaque proteins, and these cadherins that actually do the linking or the joining together. So these, like I said, are not nearly as tight. Things can get in between here, no problem. But they are good attachment sites for cells that are adjacent to each other. And again, especially in an epithelial sheet, these desmosomes are important for distributing that stress, that physical and mechanical stress, across a wider part of the tissue, rather than just being focused right there, where my finger's pointing. That stress is actually being distributed across a much larger part of my hand, which is partly why your skin doesn't fly apart every time you kind of push down on it. So these are very, very important junctional complexes within an epithelial sheet especially. There are other versions of it, so we have, again, very, very good microscopic evidence. Again, we have intermediate filaments and this large plaque aggregation, and then we have cadherins in between. There's also a sort of half version of this. So desmosomes are where two cells are linked together. Sort of a half version of this is called a hemidesmosome. Think hemisphere, half of a sphere. So this is a hemidesmosome. It's the same thing, intermediate filaments, plaque, and cadherins, except in a hemidesmosome situation, those cadherins are interacting with the extracellular matrix rather than another cell. So these you usually find, as you see here, on the bottom part of the cell. So we typically see desmosomes sort of on the side, and you see hemidesmosomes usually on the bottom, linking the cell to the extracellular matrix on the bottom, that basal lamina that we had seen earlier. That's kind of what they're looking at here. So it's called a half desmosome because there's only half the picture, basically. Right? In the real desmosome, we have intermediate filaments, plaque, and cadherins coming from both sides. Here, since we're attaching it not to another cell, but to the extracellular matrix, you only have one half of the whole picture, sort of. Does that kind of make sense? Okay, so that's a junctional complex that uses intermediate filaments. A second type of complex that uses actin filaments is called an adherens junction. Same general idea, except instead of intermediate filaments, we have actin. Instead of that large plaque-like thing, we have smaller groupings of proteins, I should say, that are called catenins. And then we still have cadherins as the hooks. Okay, so the cadherins are the same, but instead of that large plaque, we have catenins, and we swap out intermediate filaments for actin filaments. So these are called adherens junctions instead of desmosomes. And these, since they are involving actin, typically have something to do with moving in sort of the broadest sense of that term. So in certain cell types, you'll see a band of these around the cell as well. It's sometimes referred to as an adhesion belt. And this is often very helpful during embryonic development, as the embryo is sort of starting to take shape. This adhesion belt can sort of constrict up here and change the shape of the cell, so something like this. So it goes maybe for something like that to something more constricted there because of that adhesion belt kind of idea. Picture of that. Yes. As the nervous system begins to develop, the tube that will eventually become the notochord, and your vertebral column is sort of pinched off from the overlying epithelial sheet. That sort of pinching, that action here, is the result of adhesions junctions as well. So it does play a role during embryonic development as well as some other situations. And the half version of this is called a focal adhesion. So we don't call it a half adherence junction. It's called a focal adhesion. And this we've talked about generally before as well. This is what is going to allow a cell, as it's moving, as it's crawling along, amoeboid movement that we talked about last week. Obviously, the cell has to make contact with whatever it's crawling across. Those contact points are called focal adhesions, and they're essentially a half adherence junction. Actin-components, catenins, adherens. So it's similar in design to a hemidesmosome, except this one is going to allow the cell to make contact with the extracellular matrix or whatever it is that it's crawling over, and allows it to make contact so it can kind of crawl across whatever it's crawling across. So that's the half version of that. So we typically only see focal adhesions in cells that have the ability to crawl, that have amoeboid movement. So far so good? Making sense? Okay, so we have tight junctions, a very, very tight permeability barrier. We have desmosomes that sort of connect cells together in the sheet a little bit more loosely. The half version attaches the cells to the extracellular matrix. In some cell types, we have these adherence junctions, especially if you have to sort of rearrange the epithelial sheet. That's really helpful. And then in cells that crawl along stuff, we have these focal adhesions, a half of an adhesions junction. And then our final type is called a gap junction. And this has much less to do with keeping the cells sort of physically attached per se, although it obviously does, but these are much more about communication and the sharing of small molecules. So the major protein here in a gap junction is called connexin. We need to be careful here. Individual protein is called connexin, IN. And let's count. 1, 2, 3, 4, 5, 6. 6 connexins get together to form I'll go like this, connexon, ON. Okay, so connexin is the individual protein. 6 of them get together to form a connexon. So it's IN versus ON. Okay, so be careful. You can keep that straight. And then a connexon from one cell lines up with a connexon from the cell next door. And as you can see, the way they're sort of put together is that it creates this channel or this pore between the two cytoplasms of the two adjacent cells. So they make this direct connections, sort of direct pore, across the two cells. And so that small molecules can go from one cell to the other. And unlike virtually all the other transport kinds of things and pores that we've looked at, this one is very non-specific. So most of the other ones that we looked at were very specific, right? It only moved glucose or only moved histidine or something like that. This is a very non-specific channel between the two cells. The major restriction is size. So there's a size cutoff. What is that size cutoff? I think it's 1,500. Yes. So the size cutoff is about 1,500 molecular weight. So 1,500 molecular weight and smaller, sort of free access from one cytoplasm to the other. Anything bigger than that really doesn't get across very well. So what does that mean? It means basically all those monomers that we've talked about are below 1,500. So amino acids, individual monosaccharides, individual nucleotides, things like that, are all free to diffuse from one cell to the next. So we call that, that's usually referred to as metabolic coupling. So the two cells are able to share metabolites. And the other big thing that is exchanged between the adjacent cells are ions. So ions are way smaller than 1,500 molecular weight, so they can share things, sodium chloride, magnesium, whatever. All of those are small enough to cross very, very easily. So it's a way to sort of share resources among cells within a tissue. And some of these ions, as we'll begin to see more when we get back from break, are signaling molecules. So if something happens to one cell, it can signal other cells that it's attached to via an ion kind of signal that it's usually a negative signal, something's wrong, something bad is happening. So it's a way to quickly disseminate that kind of information without each cell needing to be told that individually. And so it's a way to sort of share information as well. So there are usually many of these gap junctions between cells. So you can kind of see them here in kind of cross-section. There, so usually lots of them. And calcium seems to play a role in, well, actually, let's do this one first. So again, here's the size cutoff idea. So we have several cells that are connected with gap junctions, and we inject large molecule, large meaning larger than 1,500 molecular weight, into the cell. It stays in the center cell because it's too big to get across. However, if you inject a small molecule, smaller than 1,500, into the center cell, it will cross through the gap junctions and get into the other cells as well. So it's clearly a size kind of setup. And this other experiment over here is showing that calcium plays a role in whether or not these pores are open or not. So they do have the ability to sort of shift their shape a little bit and basically close off the channel. And that seems to be controlled in part by calcium ions. And that's shown in this experiment. So again, if we inject the small dye into the middle cell here, and there's no calcium around, then that little dye molecule will get across the gap junctions and get into all 5 cells. However, if you inject a dye into the middle cell and then inject large amounts of calcium into the 2 adjacent cells, the dye stays in that middle cell, presumably because the calcium triggers the connexin to change shape, and it closes off the gap junction, and the dye molecules can't get across anymore. So calcium seems to have a big role to play in controlling whether or not these connexons are open or not, and the gap junction is intact or not. And we'll see next, or when we get back from break, that calcium often acts as an intracellular signaling molecule. So we'll see a couple of examples of that when we get to cell communication when we get back from break. Okay, so more on calcium in another week or so, but it does seem to be involved in whether or not these gap junctions are open or closed. Okay, so that's a little bit about these types of junctional complexes. So I'd like you to know, basically the function of each one, what the major proteins are for each one. Okay, so including for tight junctions, connexin for gap junctions, those kinds of little factoids. Okay, so we won't start a new topic today. Okay. So, have a great day. All gotten this far, we've all deserved a week off.